This chapter is called the physiological basis of behavior, and certainly if you've taken a physiology course, you're well aware that nervous systems are part of physiology, and so there's a way in which this is kind of a weird title, given that we just talked about the neural basis of behavior in the last chapter. But this chapter is about trying to broaden our scope a bit. So we're thinking about nervous systems, but we're also thinking about hormonal regulation of behavior and other aspects of how do we regulate behavior within an animal. And in particular, it often seems to be, as I view the chapter, focused on how do animals make switches in behavior or change their behavior depending on the conditions that they happen to be in. So how do they schedule their behavior? So actually our first topic is going to be looking at circadian rhythms and circannual rhythms and trying to understand how is it that animals schedule their day-to-day lives and what are the processes that underlie those rhythms that we all have. Then we'll also look more specifically at hormonal regulation of behavior, and we'll sort of finish with some sort of specific discussion of two major axes that are really important in terms of governing behavior. And these are the hypothalamic pituitary gonadal axis. So you see gonad in there. So this is sort of our sex steroid axis that regulates behavior. And then we'll also look at the hypothalamic pituitary adrenal axis, which is particularly responsive to stress. And so in that way, I think is a really interesting axis. And these are relevant to people just as they're relevant to at least most mammals, most vertebrates, frankly, have similar kinds of actions going on here. Okay, so if we think about this first topic, one common need that animals need to do is they need to schedule behaviors to occur at specific times. So if you think of an animal as having a particular behavioral repertoire, it can do all kinds of things. It can go foraging, and it can sleep, and it can look for mates, and it can do all these different kinds of things, but it needs to do them at appropriate times for them to be adaptive sorts of behaviors. So for example, crickets, which we've looked at a little bit, these guys need to, generally speaking, hide during the day, and they need to be active at night. So how is it that they go about setting those kinds of behavioral schedules? I think there's two major immediate proximate hypotheses that we could use to try to explain how is it that they set these schedules. One is that they could just be paying attention to environmental cues. So if they need to be silent and inactive and hiding during the day, well, if there's lots of light around, maybe that shuts down all their other behaviors and they basically just go dormant during that time. And then once there's no light around, then they become active. So this would be using strictly environmental cues in order to regulate your behavior. An alternative is that they may have some kind of an internal biological clock. So in this case, they would have some kind of a system that probably has some kind of molecular basis that occurs in a cyclical fashion over the course of a day. And so even in the absence of environmental cues, you would still expect them to cycle in these cases. So we have two different explanations here, and I guess they're not really mutually exclusive, but they do kind of set up two alternatives, and maybe the truth lies with one or the other or somewhere in between. So this is kind of my question to you guys is, how could you tell between the two of these? What could we do to try to do an experiment to test between these two hypotheses? And you already have your hand, so yeah.
You could take some crickets and keep them in a controlled environment, like a constant temperature with ample food supply.
Mm-hmm.
Either like constantly in the dark or constantly in the light.
Right, okay. Yeah, so if they're responding to cues from an environment, if that's one of the hypotheses, and then we remove all cues from the environment, so always dark, steady temperature, no changes in noise ideally, which, I mean, to some extent it becomes very hard to do these kinds of studies to remove all cues, but certainly, removing all light cues, that's pretty easy to do. You just leave them in the light all the time or leave them in the darkness all the time. And then you see what happens. So we predict that if they're strictly using environmental cues and we shut down all environmental cues, then they would cease to cycle. They would just kind of have the same behavior all the time, be it always hiding or always active, depending on if the light was there or not. Whereas if they have an internal biological clock, I think we'd expect them to keep cycling. Right? So here's an experiment that did just that. So in this experiment, the way you read these figures, they take a second to get a handle on, but they're not too bad. So each of these little lines right here is one day, one 24-hour day, and it even shows from 0 to 12 hours to 24 hours. So this is midnight, and then on the other side over here is midnight of the next day. And so this is following one individual over the course of, about 10 days in the first part, and then that same individual for another, 20 or so days in the second part. Again, each of these horizontal lines is a day. I guess each of the vertical lines is probably some fraction of an hour. And then what they do in the case of crickets are particularly attractive for this kind of a study because you can automate a lot of these processes. So I've seen different kind of rigs. Sometimes they have like some kind of sort of a force sensor in the bottom of a little cage, and they'll have little cages for crickets, and they can do dozens of animals at once, which is kind of nice because they don't need a lot of space. And so every time the animal walks around, it sort of records some action. In this case, I think they were actually recording chirps. And so they were only recording chirps at such times that what I should say is they had a little microphone embedded in each of the little cages for each of the little crickets. And so here we're observing each time there's a little bump, like is seen here, that's falling off of your main line, that's indicative that there's some kind of a chirping activity. But you could just sort of think of these sort of blotches that you're seeing up here as animal is active during this time. In the first experiment, they have taken an individual that was on a normal light-dark cycle. So maybe it was 12 hours of lightness and then 12 hours of darkness. And they've thrown that individual onto a constant light cycle. So that's why the sun is sort of shown throughout this period. And it's light, 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 light all the time, all day, all 24 hours. And so, I don't know, what do you guys see? So here's this first day, here's the second day. Do you see them continuing to cycle on a daily basis? What do you think? What do you think?
They're still cycling, but
yeah.
The cycle is influenced by environmental cues.
Why do you say it's influenced by environmental cues?
Because it's kind of shifting to
yeah.
right, but it's still like a consistent length.
Yeah, so it's roughly the same length every day. I mean, it gets a little broken here and there, but it's roughly, about that long. But each day it seems to shift a little bit further back, so they're not getting up and starting to become active at the same time each day. There's a little more of a variation that's occurring, such that they seem to be clock shifting back every day. And so you're seeing this same animal. You can see at this point it's starting to shift so much that its activity is bleeding into the next day. So that's why we're kind of seeing these notches that continue on to the next day. What they do is they take this individual once it gets to this day. On the next day, they shift it back onto a light-dark cycle. And so here's 12 hours of light and then 12 hours of darkness. And you can see it takes a couple of days for the animal to entrain, and that's the word we call it entrainment. It takes them a couple days to entrain to this light-dark cycle. Effectively, the animal's kind of jet-lagged at this point, and it takes a couple days to get back on the normal cycle. But at that point now, it's really consistent. As long as it has this light-dark cycle, it gets up at the same time every day and then stops being active at the same time every day, coinciding with the light-dark cycle that they are existing on. So yeah, I would agree with you. I think clearly there's an indication that they do have the ability to cycle, which means there's got to be something internal that's going on, right? There's got to be some kind of an internal clock existing for these guys. But at the same time, there's got to be some role in environmental cues, because once you add the environmental cues, then they have the ability to cycle in a very consistent manner, and they kind of entrain on that light-dark cycle every day. So what is the mechanistic basis for this rhythm that these crickets seem to have? You can do experiments where you can just start playing around with their nervous system and trying to figure out, all right, what parts of their nervous system are important for this thing. So as we've seen, crickets have an internal biological clock. On its own, it cycles about every 25, 26 hours as opposed to every 24 hours. So it needs some kind of environmental cues to entrain it. There's that word entrained. And humans actually have roughly the same kind of a cycle. So if you take a human, and people have done these studies where you basically find a really low subterranean environment. Oftentimes you find a cave or something where you can not bury this person, but basically get them separated from all cues. You keep temperature constant, you keep noise constant, you keep light effectively constant, and you remove all clocks, all other stimuli, and you just kind of watch what the person will do. People will cycle on roughly about a 26-hour cycle. So they'll stay up a little bit later every day in the absence of any kind of specific cues telling them to do this otherwise. And crickets seem to do the same thing, as do apparently most animals. This is one of the reasons why with shift workers, the recommendation is that if you're switching shifts, like if there's 3 shifts during the day, that it's better to shift to shift forward, so you're shifting to the later shift. So you just stay up later as opposed to trying to get up earlier, because our natural tendency, if anything, is to stay up a little bit later each day, as opposed to go to bed, or as opposed to getting up earlier each day. Anyway, that's a sort of an aside. If you start doing experiments where you start removing different parts of a cricket's nervous system, and you can do this just to experiment and try to figure out where is this clock existing within the crickets, what you find is that certainly if you cut neurons coming out of the eye itself, out of the compound eye, the animal loses its ability to entrain. So it's definitely receiving visual input from its eyes and using that information to entrain on whatever the current light-dark cycle is. However, so you could sort of imagine maybe the retina of the compound eye is out here. But this structure in here, this little bulb, this is called the optic lobe. So this is not actually part of the eye. This is basically the part of the brain that's receiving information from the eye. And if you remove the optic lobe, not only can they not entrain, but they just cease to have any kind of schedule or rhythm at all. And so apparently, at least within crickets, somewhere within this structure are cells that are either by talking to each other or having some kind of an internal process, they are determining the circadian rhythm of these crickets. So here's kind of a flow chart that your book puts forth that tries to explain the cricket circadian rhythm. So the idea is that there is sunlight or some other environmental cues. Clearly, light-dark cycle is really important in terms of determining the circadian rhythm of these crickets based on that experiment we just saw. But those are received by sensory receptors. In this case, these would be receptors in the eye. And they're fed into a clock mechanism, which includes a pacemaker. And so the idea with this pacemaker is that this is some kind of an internal system. And you saw an example of this in mammals on the homework, which we'll talk about. This pacemaker must exist somewhere in their optic lobe. But it's setting a rhythm for the animal. And then that pacemaker, either directly or indirectly, talks to other brain systems that govern locomotion patterns. So this is basically activity. Feeding behaviors, hormonal release patterns. Hormones will fluctuate over the course of a day. And other kinds of rhythms that determine and cause the animal to have some kind of a daily circadian rhythm. So again, on the homework, there's the discussion of that SCN, the suprachiasmatic nucleus, and some of the genes that seem to be housed within that structure that are functioning as a pacemaker. So we'll kind of come back to that when we get to the homework as far as trying to understand what is the mechanism here. But at least I wanted to sort of present this one example of circadian rhythm. Any questions on crickets and circadian rhythms? Mm-hmm.
So by cutting out the neurons, is it that they're not receiving the sensory information, which is why they use entrainment, and then the optic lobes that they're not getting the messages that they're
Yeah, so this first bit where you're cutting neurons from the eye, you're just losing the sensory. So you're cutting here, and you're not getting to the pacemaker. If you cut out the optic lobes, at least in a cricket, the optic lobes are parts of the brain that are in charge of processing visual information, but they seem to be the location of the pacemaker. So if you cut out the optic lobes, you lose all rhythm altogether. Not that they won't entrain, they'll just be active at random times and lose any kind of a rhythm to themselves at all. It's not unlike there's one of the fruit fly mutants that was on the homework also. Good question. You good with this? The other kind of rhythm that we'll see across animals that I think is also really interesting and is important in determining important events that occur over the course of the year are what are called circannual rhythms. So animals often have to cycle in their behaviors over the course of an entire year as well. Some of the major key events that happen in an organism's life have to happen at certain times of year. So they need, if it's a bird or a mammal, they need to molt or shed at certain times of year. They need to enter into a reproductive state oftentimes. Most of the time animals, unlike humans can kind of reproduce all throughout the year. Maybe they're on a monthly cycle, maybe not. But again, humans can kind of reproduce any time. Most animals are only available only available to reproduce once or twice a year. And it has to happen at a certain time when environmental conditions are optimal for raising young. Because if you try to reproduce at some other time, you're not going to have a successful clutch or brood or set of offspring. So behavioral cycles need to cycle over long periods of time over the course of a year. And we do have annual cycles of reproductive bouts, of hibernation. There are all kinds of important behaviors that have to happen on some kind of a rhythm. So again, I think we could ask, how is it that animals determine when to start into a reproductive state, or when to start into, going into a hibernation or a dormancy or a torpor kind of a state? And we could sort of raise the same two hypotheses. So we could say, well, maybe animals are responding to cues from their environment, or maybe they have some manner of an internal circannual biological clock that determines their annual rhythm. Which, I don't know, like from a molecular basis, I can kind of get how a sort of a daily clock can work within an organism. I think it's kind of amazing to think that maybe they might have some kind of an internal annual kind of a clock that somehow builds over the course of 365 days. And that has to happen at a molecular level. And I think that's kind of amazing if that's true. But it's certainly possible, and we could raise the same kind of an experiment where we remove any kind of environmental signal, so we don't allow for changes in the day and the night cycle, which typically do happen at different times of the year, at least in northern or southern hemispheres away from the equator, where you see substantial differences in light-dark cycle. We could prevent any kind of changes in what other kinds of cues. Temperature, certainly, rainfall, these kinds of things. And we can see, once you remove all environmental cues, do animals still cycle? And the answer is that in many cases they do. So here's a little chipmunk. So chipmunks, these are little ground squirrels. They alternate between a hibernation state and an active state every year. So during the winter they hibernate, and then they come out during the spring and then stay active out until the fall. They're starting to kind of get their stores up and they're just approaching the time when they're going to start hibernating. I see them on roads a lot right now. So here are 5 animals that they did this experiment on. 1, 2, 3, 4, 5. And then here on the x-axis is years, and the bar indicates when they're hibernating. And I think in this case what they did is that they have these animals, instead of observing them in the field, they're observing them in a laboratory where, again, they've removed any kind of environmental cue. And lo and behold, despite the removal of all environmental cues, they keep cycling. Every year they go through a hibernation state. You can see that initially, this first year, when all the animals had been on sort of normal environmental cues, they were all cycling at basically the same time. As the years go on, they start to kind of all get out of phase with each other. So there's this one guy, 3, that keeps hibernating a little bit earlier each year. Some of these guys have maybe shifted a little bit further back, or I don't know, maybe they're just more on rhythm. But they're all continuing to cycle. So there's got to be some manner of an internal biological clock that determines circannual rhythms in addition to circadian rhythms. Here's another example. This is a case of a bird. This is a stone chat where they were doing the same deal. They're just basically following this guy every year. And so in this case, each of these graphs is an individual year. They're labeled, this is 1982, this is 83, 84, 85, 86. And what you're doing is you're observing three different things on this figure. One is when they're molting either their wings or their body feathers. That's something that these animals need to do on an annual basis. And in addition, you're also observing testicular width. Birds, like a lot of other animals, will kick on their reproductive systems at certain times of year and then turn them off at other times of year. And there's a variety of reasons for this. But one thing that seems to be true is that as you increase testicular width, you tend to increase testosterone levels. Testosterone levels tend to cause animals to become a little bit more aggressive, at least these particular animals. Causes them to be more territorial, and therefore they're spending a lot more energy on territoriality and aggression, and potentially imposing some risk on themselves because they're getting, effectively, I just tend to think that animals on testosterone tend to be stupid. And so they might be taking on, more fights than would maybe be optimal in terms of survival, at least. It might help them with reproductive opportunities, but it might be bad for survival. But also, testosterone tends to impair one's immune system function. And so, again, you just don't want testosterone circulating around all the time if you don't need it all the time. They really only need good testicular width when they need to be gearing up for reproductive season, producing sperm, having good testosterone levels so that they can be aggressive and secure territories and such. So they will alternate pretty dramatically over the course of a year. So again, what they've done in this case, we can measure those three factors. We can measure when they molt, we can measure when they have surges in testicular width, and then when they recede. And then in this case, they also removed all environmental cues from these animals. So the question up here I think should be straightforward. Do these data support the environmental hypothesis or the biological clock hypothesis? Again, these animals have no environmental cues. So what do you think? Environmental, biological clock. Do they support the biological clock? Let's start there. What do you think?
Looks like it does, at least maybe with like testicular. It looks like it's cycling for the most
Yeah,
part.
they keep cycling. I think I would agree with you. I mean, I think they do cycle. This little dotted line they kind of imposed here sort of shows how they do shift a little earlier each year.
Kind of changes. I know the width of the molts fluctuate a little bit.
Yeah. But they still do them, right? So, I mean, I think there's still argument here for a circannual rhythm, but then clearly, again, these guys this individual is no longer doing it at the same time every year like it normally would, so there's an environmental component as well. There must be, because they need to entrain these rhythms. Okay, so what then are they using to entrain these rhythms? What are the cues that, I mean, these are two examples with the chipmunks and with the stone chats where they have no cues. So what kinds of cues are able to entrain these kinds of rhythms? By far and away, I think the most common is photoperiod. I'm going to turn the figure up, or the lights on a little bit, just so you don't fall asleep. So we're going through a period where our days are getting a little bit shorter every day right now. And I tend to be, my daughter started junior high, so we are now getting up at 6 in the morning every day, which, oh my God, that's brutal. I mean, it's not going well for me. But the thing that clearly has been noticeable is that when school first started, 6 a.m. sucks, but like there's some light outside, right? And now, 6 a.m., there is no light outside when we get up. It is pitch black out there, and it doesn't really start to have any kind of light till around 6:30. I went for a run this morning at 6:30, and I've got like flashers on it. Pretty soon I'm going to need a freaking headlight, or else I'm going to run off the road because I won't know where I'm going. And that's occurring on the mornings, but it's also occurring in the evenings. So we're going to consistently have earlier dark times, and you can observe this over time. And animals seem to observe this too. So there are all kinds of interesting questions about exactly how this works. There seems to be these cool systems where animals will, when they first experience light, there's some sort of a system that will turn on that will then inhibit some other thing for 18 hours or something, and then they, I mean, your book goes into this, and you can try to get into it. I find it's more trouble than it's worth. But, I mean, the mechanisms by which this work, I think, are interesting. But again, animals seem to clearly pay attention to light-dark cycle. And so as days get shorter, then that informs them that they're approaching more of a winter type of a period. And so if they're going to enter into a dormancy, or at least if they're going to molt into their winter plumage, or if they're going to start stashing away food, during this period of time so they can survive through the winter, that it sort of informs them that that is the time that they need to do it. And the animals very likely are not, I mean, they're very not likely not thinking about this. It's just that as the days get shorter, that triggers certain behaviors to start to be employed. Oftentimes, if you're looking at animals that are in captivity, and if you're trying to get them to breed, so a lot of zoos are aware of this, for example, they will require a certain light-dark cycle to permit breeding. So if you just try to keep them on 12 hours of light and 12 hours of dark all year, they oftentimes will not breed. You have to fluctuate them, take them down to a short day, let them expand up towards a longer day, and it's only then that they will be able to actually breed. Dr. Wolf keeps a zebrafish colony, zebrafish? I think that's right, colony down in the aquatics lab. Not in the aquatics lab, in the vivarium that she uses for developmental studies. But in order to get them to breed, she has to screw around with their light-dark cycle. So if she holds it constant, again, they won't breed. So animals clearly seem to pay attention to this. This is just one example. This is this white-winged crossbill, where I think in this study, these are animals that were kept in captivity, but the only cue that they allowed to vary was light-dark cycle. And you can see that their testis length did fluctuate and coincide with the light-dark cycle, so that in this sort of June, July months is in both years that are in the study is when they had maximal testicular width, and then they would degenerate down then in between, just like we saw with the stone chats. Not all animals use light-dark cycles, though. So light-dark cycles work really well if you're in an environment like a lot of temperate areas where there's pretty predictable weather at certain months. Right? So the light-dark cycle sort of tells you effectively what month is it. And if you know that, generally speaking, June and July are great times to be breeding, which seems to be true for this crossbill, then that's a great time, that's a great cue to use to inform you about when to turn on your testes and turn them off. However, there are some environments where that just isn't the case, where there's unpredictable quality of environment at different times of year. So the equator, for example, if you live in equatorial regions, they don't really have much of a fluctuation like dark cycle. The thing that matters for them usually is they tend to have a rainy season versus a dry season, and they need to plan around that. So they need to use other kinds of environmental cues. This bird, this is a rufous-winged sparrow. They live in the Sonoran Desert, which is the desert in the American Southwest. And so in that desert, there is a fairly predictable monthly cycle, but the key thing that they need is that they're looking for the monsoon season, they call it, which is basically the rainy season to start. And that can start anywhere from like mid-July to mid-August. So if they start to try to reproduce early, and they beat the monsoon season, then they're basically going to be trying to reproduce under just incredibly hot, dry conditions where there's just no food around. Whereas if they wait for the monsoon season to strike, then all of a sudden this is when the desert really comes alive. Lots of plants are produced, insects come out, there's bugs flying around everywhere. These guys love to eat them. And so as a result, that's sort of optimal time to be trying to raise offspring. And so what they do is they seem to be using rainfall as their main cue. They can pay attention to light-dark cycle, but it doesn't seem to be a very good cue for them. But if you expose them to rainfall, they will show a pretty substantial shift in a lot of their structures associated with reproduction. So this is that higher vocal center, that area we talked about back in chapter 2, that is controlling a lot of song learning and as well as song production. After rain, it increases its size pretty dramatically from about 0.4 to about 0.6 cubic millimeters. This is the RA structure, which is another one of those vocal control centers within the brain. It also shows dramatic increases in size. And then this is an indication of how many birds are singing per transect. So this is just going out, walking in a transect, noting how many birds are being active. And you can see that before rain, they're pretty much silent. But once the rain hits, they start singing like crazy because they're all trying to secure mates, get nest sites going, and reproduce, get your chicks going as quickly as possible. All right. Any questions on any of that stuff for now? Okay. So what I want to do then is switch gears a little bit. I should have had a chapter outline slide come up so we could transition. And instead of focusing on these sort of behavioral schedules, talk about other kinds of switches in behavior. So how do animals shift between behavioral states beyond just sort of circannual rhythm kinds of decisions? And here's an example that's from your book, but I think it's a good one, with infanticide in mice. If you find a male mouse, just encounter this guy without pups, they're really prone to infanticide. So if you have a male mouse, you stick him in a cage, maybe there's a female there, maybe there's not, but if there's pups present, generally speaking, male mice that have not encountered and have not mated will tend to be really prone to infanticide, and they'll just go around killing all the pups that they encounter. They're just, awful little things. And they continue to be infanticidal. Even if they get an opportunity to mate with a female, they'll continue to be infanticidal for about 3 weeks after mating. But then there's some kind of a switch that happens, where after about 3 weeks post-mating, then the males will suddenly be protective of their pups for about 4 weeks. And then once week 7 hits, they become infanticidal again. I mean, they're just, it's like they can't make up their mind and they can't decide if they're going to be good parents or not. First of all, why do you think that might be happening? We could sort of take it from an adaptive standpoint. Like, why would this be an adaptive behavior for these male mice? I'm going to argue that it is. Why would they stop being infanticidal at 3 weeks? What do you think? Yeah, so I don't know if they're realizing it, but I'm sure their hormones are fluctuating. But I think that's kind of the argument, is that maybe they don't know what's going on, but selection knows what's going on, right? So after 3 weeks is basically the gestation period of these mice, and so there is a possibility once 3 weeks hits that those mice those pups that they encounter could suddenly be theirs, and they don't know if they're theirs. They're just this is a change in behavior towards pups, but they start off being fairly infanticidal. And we talked about why be infanticidal. This may open up mating opportunities for you later on if you get rid of the current offspring that are around in your social group. But once 3 weeks hit, you don't want to kill your own pups, and so selection has favored individuals that do not kill pups after about 3 weeks. And then after week 7, at that point, your pups should be grown and they should be out, and at that point, you're no longer at risk of killing your own pups, so why not be infanticidal again? That seems to be sort of the adaptive argument, what's going on. There was a question of how exactly is it that they're doing this. So in terms of mechanistically, what's going on in their brain? How are they measuring the time that's passing here? And so this was a study that was trying to understand exactly how it is that they did measure the time. So what they did is they had individuals that, they didn't have any, I think they're not presenting any anyway, control individuals where this is sort of real time. But then they had a group of individuals that they put on slow days, where every day they had them on a light-dark cycle, but every day was about 27 hours long. So it was 3 hours longer than a regular day. And it was very predictable. Every day on this clock, the light would come on, but it was coming on after 27 hours every day instead of after 24 hours. They had another group that were on faster days. These guys only had 22 hours in their days. So we're manipulating the environment around them. They have an internal biological clock, but this is within the realm of something that they can entrain on. And then they basically just measured how much actual time has passed in terms of like the real clock that we could measure with a, with a wristwatch. But then also how many light-dark cycles have they experienced? Because individuals that are on slow days, after 16 real days, for example, the individuals that are on slow days will have experienced fewer days passing than individuals that were on the fast days. There'll be more light-dark cycles if you're on the faster day after a certain period of time. And so they basically just went then and checked, all right, so is this changing the behavior of these animals? So if they're basically just counting the number of day-night cycles, and if after 3 weeks, which would be 21 days, if that's the time when they normally would stop being infanticidal, so here's 20 days. These would be the actual days, but the individuals that are on the slow days, they wouldn't actually hit that 20 light-dark cycle until, over here. This is like 3 or 4 days later. Whereas those individuals that are on the fast light-dark cycle would reach 20 light-dark cycles. Instead of after 20 days, they might reach it, around here, around more like 18 actual days then. So here's what they tried to do is basically assess, does this alter the frequency with which males commit infanticide? And so here on day 1, everybody commits infanticide, right? It's the thing to do. So all these males are horribly infanticidal, and this is 1 day after they've mated, because at this point, there's no risk that these could actually be their pups. So from an adaptive standpoint, this makes sense. After 16.3 real days, so by our wristwatch, 16.3 real days, what they find is that the individuals that were on the slow days, the individuals that were on the fast days, why is that not making sense? I'm getting myself backwards here, but no, slow day males should take longer to do this. And this made sense when I was looking at it. Real day sense mating. We've got fast day males committing infanticide more often. That should be backwards. I wonder if the figure is wrong. Am I missing something here? I don't know what's going on here. Day 20, I think, makes sense, though. Yeah, I'm sorry. I don't know what's going on with this one. Let's look at 20, though. At day 20, this would make sense, right? So at day 20, the individuals that have been on the fast days have experienced more light-dark cycles. And so these guys are non-infanticidal at this point. They're not committing infanticide. But the individuals that were on the slow days, they're still infanticidal because they've really, while 20 days have passed, they've only sort of experienced something more like 16 light-dark cycles. And so they, in their minds, whatever that's going on with their minds, in terms of what their nervous systems have experienced, they haven't experienced as many light-dark cycles. And so for them, they're still within that window where these couldn't be their pups. And so they continue to be infanticidal when they're on longer days. I really think they just forgot to put the bar in for that figure, and I didn't notice until like right now, and I'm sorry about that. But these slow day individuals do finally stop being aggressive once you get to about day 24 of real days, because finally they've kind of gone through their full 21 light-dark cycles, being on the slower day pattern. I think the thing that really catches it is that even though we are able to induce differences in our groups by putting them on these 2 different treatments, if you look at total number of light-dark cycles since mating and then compare the behaviors of these animals, if only 1 light-dark cycle has happened, both groups are equally aggressive. After 18 light-dark cycles, whatever hourly basis they're experiencing them, after 18 light-dark cycles, both groups are pretty aggressive. But after 22 light-dark cycles, they pretty much have all stopped being aggressive. So it's the light-dark cycle that seems to be the thing that they're counting. Somehow their brain is counting how many times have they gone through a dark versus a light period, not the actual hours that have passed. That doesn't seem to be what they're tracking. I'm convinced that there should be a red bar here that's like up to here, and it's just, I didn't notice it till now, so I'm sorry about that. Does that make sense? Do you have questions on it? I'm going to have to pull the original study and get the figures from the original study next time. Okay, so we've got a case where animals are shifting their behavior. That's the big picture point here. And they're doing so with some kind of a thing where they're counting light-dark cycles. The next question would be exactly how is it that that's happening? And you had mentioned, Alexandria, possibility there might be hormones that are fluctuating, and so there's some sort of hormonal difference between these animals, and I think that's a pretty reasonable hypothesis. So here's a figure or a graph where we're actually trying to assess what hormones do differ between inexperienced males and fathers. So inexperienced males, these would be males that have not mated before, or it could be probably males that have just mated but they don't have pups yet, versus individuals that are actually fathers, and therefore they've passed sort of this 3-week mark where there are pups on the ground that are theirs. And here are two hormones that they're assessing in this case. This one's testosterone, which that's kind of a natural one, I think, to look at, right? This one's progesterone, which is kind of interesting. Progesterone, I think, is largely thought of as one of the hormones involved in female reproductive systems, and it is. It's the hormone that maintains the uterine lining, for example, during pregnancy, and when this level drops, this is what induces a menstrual period in human females, at least. So this, that certainly plays that role, but here they're assessing whether or not that might play a role in regulating the behavior of these male mice. So the mated without pups bars, I think, are a little bit confusing, but again, these are guys that probably have just mated, but they haven't had time that their pups wouldn't have been born yet. So in any case, I think the interesting bars to compare are the inexperienced males, so these are basically the virgin males that have not had the opportunity to mate, versus the fathers, which are over here, which are individuals that have had the opportunity to mate and do have pups already. And if you look up there, I don't know, what do you see for testosterone and what do you see for progesterone? Do you see differences across them? Or where do you see the biggest difference? And again, I ignore this bar for now because it gets a little bit confusing. But, if you compare, so again, here's sort of the virgin male versus the father male. And I don't know, like this testosterone bar, it's a little bit lower. The error bars, if we were to fold that error bar down here, it would, almost be touching this one. I'm not even sure if these are significantly different. They may be, but it's not a huge difference in terms of testosterone titers between the inexperienced male versus the father. But look at progesterone. Progesterone, inexperienced males have really high progesterone levels. And the fathers have very, very low progesterone levels. And again, if you kind of fold this error bar down, it would only come down, this one would only come down to maybe about here. Trace that over to here. Clearly there's no overlap between those bars, and those are significant differences. It turns out that based on these and other data, it seems as though progesterone seems to be a really important determinant of behavior in mice. So on the prior slide, when we're talking about these mice males changing their behavior and switching from being infanticidal heathens to these sort of nurturing fathers, the thing that seems to be most different in those individuals is that their progesterone levels have really, really dropped. And we can test this further. We could say, all right, so if we think progesterone is the thing that's causing them, in this case, to be infanticidal, and in this case, to be nurturing, loving fathers, let's try screwing around with the progesterone receptors. And this is the great thing about mice, is you can use genetic methods to knock out the genes that encode for progesterone receptors. What would you expect to happen in this case? So if we knock out the progesterone receptors, so the animals lose their ability to detect progesterone levels, what should that do to infanticide, do you think? So if they no longer can detect progesterone when it exists, when it's being produced, it would almost be like progesterone was never around. So what should that do to their behavior? Ideas? I like to think about predictions before we actually look at the figure so we know what we're looking at. So in a knockout experiment where you knock out the receptors, progesterone levels may be super high, but we are making it so the cells, the neurons of these mice can no longer detect progesterone when it's present. So effectively, we're making everybody kind of like this, right? Because we are removing progesterone from the picture. We're removing the ability of their neurons to detect progesterone, even if it's there. They can't detect it anymore. So their cells should respond as if there is no progesterone present. And if we're right that progesterone is what's causing infanticide, then if we eliminate any function of progesterone from the brains of these mice, then these mice should really stop being infanticidal. And that's exactly what happens. So here are control mice. They did 2 different litters to make sure there's sort of no history effects, but these aren't meaningfully different from each other. But these are control mice, and they're pretty infanticidal. These individuals are the progesterone knockout mice, though. And so in this case, when you knock out the progesterone receptor, you knock out the ability of these mice to actually be infanticidal. They just, all of a sudden act like wonderful, delightful fathers. So it's a dramatic effect, right? I mean, it just wipes out infanticide altogether, and it all has to do, apparently, with this hormone that's necessary for infanticidal behavior. Kind of makes me want to figure out a way to knock it out in all males in humans too, and just see what happens, but probably be unethical, I guess. So the idea here is that this hormone, progesterone, is having what's called an activational effect on a behavior. In this case, the behavior is infanticide, and when progesterone is present, it activates the infanticidal behavior. There's some other ways that hormones can affect. They can have sort of more of a developmental kind of an impact where they change developmental trajectories. Here we're just focusing on what your book defines as an activational effect, and that's where most of the time we're going to be. Let's take a 5-minute break just to let you get your thoughts. So I got 10:25. Let's resume at 10:30, and we'll finish off these slides and talk about a case where hormones can activate certain behaviors that hadn't really existed before. So this is asking, why do quail stare at their partners? So it's sort of a fun little experiment you could do with some quail, where you can have a male housed next to a female quail. And here are these little quail. They're cute birds. And before they mate, the male will occasionally, vocalize or stare at a female, or look, glance through a little window in this case. So they've got a little window where he can watch a female over here, and the male is over here. And he will do this occasionally, but he won't spend very much time doing this. But after you let them mate, he will basically just stay there staring at her constantly all the time. He's just really, really entranced with her. So you could ask, why is he doing this? Why has there been this shift in his attention that he's paying towards a female partner post-mating? And so one argument I think you could put forth is that there's some kind of hormonal surge. So maybe he's got a surge in testosterone levels, for example. So they did an experiment where it's kind of a rough experiment on these poor guys. So first they would castrate the males. And then they would give them implants of testosterone to some of them, and then they would give no implants of testosterone to the other ones. And so in this way, then, they could observe whether or not the change in testosterone that they're experiencing is affecting the staring behavior of these animals. I do want to ask, I think it's kind of an interesting system. Why do you do this and instead of just castrating a few of them and leaving others with intact testes? Like, why would you castrate everyone? These poor things. Yeah, go ahead. Yeah, Alexander. Yeah, is that what you're going to say, Kristen? Or yeah, so I think this is the idea, is that you want to, this allows you to be a bit more controlled. So testosterone, the testes can produce testosterone, but you don't really have control over how much they're producing. Whereas in this case, you get to directly regulate how much testosterone these individuals are experiencing. So they do this experiment, everybody gets castrated, and then they've got implant males versus other individuals that they get an implant, but it basically just has water in it, so they don't get the testosterone at all. And here's what happens. So they're measuring time at the window. They just, get a stopwatch and they watch how long over a period of time these males will spend staring longingly at their mate. And in the individuals, so here are, there's 3 different groups here. There's testosterone implanted. There's another one that we'll get to in a second that's testosterone implant plus aromatase inhibitor, but don't worry about that for now. And then there's controls, and I'm having a hard time seeing the differences in color, so I'm going to turn the lights down. So the controls without the testosterone implant clearly just don't spend much time at the window. So if you wipe out someone's testosterone, this is indicating that it does wipe out this behavior. Compared to the red line, I'm sorry, the blue line, which are these guys, where these guys do get the testosterone implant. They get a testosterone surge as a result, and it's regulated, so they get this surge. And they do then spend lots of time staring longingly through the window. And they continue, it kind of ramps up through the first 9 tests, and then they kind of, I don't know, they sort of plateau, kind of. Maybe they even still keep on going. I don't know. There's still a little upward trajectory, I feel like, in those data. They're spending lots of time at the window. And so here testosterone seems to be having an activational effect in inducing this staring behavior. Now the one that's kind of really interesting is the red line. So the red line is exactly the same as the blue line, both in terms of the response, but also in terms of what they've experienced. They all get testosterone implants here. But at test 9, the red line individuals get an aromatase inhibitor added into their system. And this aromatase inhibitor, once that happens, you can see what occurs is it basically knocks out the staring behavior and turns off the staring behavior in these animals. So then, of course, the question is, well, why did they do this? What's an aromatase inhibitor? So here's the cool thing. The way testosterone actually works in a lot of cells is that testosterone actually enters into cells, a target. So here is the cell membrane of a target cell. Here's testosterone. It passes through the membrane because it's hydrophobic, enters into a target cell, and then is acted upon by an enzyme in the target cell that's called aromatase. And aromatase converts testosterone into estradiol. Estradiol is like one of our main estrogens. And then the estradiol can bind to estrogen receptors and then interact with things in the nucleus. So curiously enough, many times the actual effect of testosterone, where we think of testosterone as causing certain effects on individuals, is actually mediated by not by testosterone directly, but instead it has to get turned into estradiol, and then the estradiol in turn does the actual effect on the target cell. The enzyme that does that conversion is aromatase. And so in that experiment, if I go back one, whoops, these guys in red, right here, they got an aromatase inhibitor. So at this point, they no longer have the ability to convert testosterone to estradiol because you've inserted an inhibitor that knocks out that enzyme. And so, and you can see what happens is once they no longer have the effects of getting estradiol produced from their testosterone, they lose the ability to induce and activate that behavior. You're basically turning them into what looks like a castrated male. By the way, somebody asked once, I have no idea what's going on with this green surge here, where just all of a sudden they get interested. Yeah, bless you. No idea. Behavioral data are noisy. So I think this is kind of a neat little story. It does show a nice example where a hormone, maybe a pair of hormones, are activating a behavior. It also shows that many times what we think of as testosterone's effect is actually mediated by estradiol. So many of testosterone's effects are actually driven by estrogen receptors within the target cells of the animal, which includes the staring behavior. Questions make sense? Be it testosterone or estrogen or, frankly, also progesterone, all of those are gonadal hormones. Get the lights back on. And so all of them are produced, and this is just sort of like a crash course in a little bit of endocrinology. All of them are produced via this hypothalamic pituitary gonadal axis. So they're showing this in humans, but this is basically true for most vertebrates. I'm not sure on fish hormonal control, for example, but certainly any reptiles, birds, mammals, anything within that group, they all do this kind of have the same kind of a pattern. So the way it works, the reason it's called a hypothalamic pituitary gonadal axis is that it's not just like one gland is acting by itself within the organism and producing a hormone. Instead, it's this sort of like cascade of hormones that are released, and this cascade is called an axis. So here they're kind of zooming in this one little region of the brain, and sticking off the bottom of this brain is this really important region called the hypothalamus. The hypothalamus is part of, it's not quite midbrain, it's called diencephalon technically, but it's sort of central within the sort of interior part of the brain here. But it is one of the main regulators of our entire endocrine system. And the way it works is it releases what are called releasing hormones. In the case of this particular axis, it's called gonadotropin releasing hormone, which travels from the hypothalamus down to the pituitary gland, which is just this little, looks like a little punching bag hanging off the bottom of our brain. The pituitary gland, in turn, then gets the releasing hormone, and the releasing hormone causes the pituitary gland to release its hormones, which are, in this case, luteinizing hormone and follicle stimulating hormone. I should have written those out, but I didn't. Let me write them on the board. So these are pituitary hormones, and their job is to travel down to the gonads Sorry, I can't talk and write at the same time. So we've got luteinizing hormone, LH, and we've got follicle stimulating hormone, FSH. Their job is to get into the blood, and they travel from the pituitary gland down to the gonads, be they testes or be they ovaries in the female. In either case, they're going to cause these glands then to start releasing their own hormones. And so in the case of the testes, the main hormone that will be generated would be testosterone. In the case of a female, the main hormones that would be produced would be estrogen and then also progesterone, depending on where they are within a given monthly cycle. And these hormones will typically and commonly induce reproductive behavior and development. So testosterone, for example, in males triggers all of our secondary sexual characteristics. So in people, we're talking about, broadened shoulders and, broader foreheads and deepening of voice and, everything else that goes along with sort of being male. And similarly in females, when you have estrogen and progesterone release, especially once a person hits puberty, it causes all the secondary sexual characteristics that are associated with being female. So they certainly cause all those kinds of things, and they kind of prime up a reproductive system. Testosterone is involved in sperm production. Estrogen and progesterone are involved in the development of and release of eggs every month for females. These hormones can also cause behavioral changes. So testosterone oftentimes, in certain individuals anyway, can cause aggressive behavior. It's not a universal thing, but oftentimes that is true depending on species. And those aggressive behaviors may be involved in securing territory, securing mates, and performing well in their given environments. It is worth noting, I kind of made an allusion to this before, but testosterone also can be pretty harmful. Here's an experiment where they're looking at these little guys. These are side-blotched lizards, and they're a fairly common lizard. I think these are from the American Southwest. But these guys, you can't really see from here, but if you pick them up and look at their throats and their bellies, they have like a big colored splotch, kind of like right through here. And a lot of lizards have this kind of appearance, especially within this particular lineage. Here, what they did in this case, this is just sort of like a grab and stab study where they went and sampled individuals that had one of these three different throat colors within this one species. And you do see all three morphs within this one species. You see yellow males and blue males and orange males, and they measured testosterone in each. And what they found is that the orange males, the males that had orange throats consistently, had higher testosterone titers than the others. Interestingly enough, these guys also tended to be a bit more aggressive. They tended to be a bit more successful in securing territories. So there are some advantages with being orange and having this high testosterone. However, when you track survival into the next year, so you just sort of measure how likely is it that yellow individuals or blue individuals or orange individuals will make it to the next year, and you control for their actual age also, here's what you find. Yellow individuals tend to survive, about 25% of them survive to the next year. That's decent for these animals. Blue individuals are the same, but orange individuals, God, they just don't make it, right? I mean, there very few of them are actually surviving through to the next year. The argument is that the two things are going on. One is that these, because they're so aggressive and engaging in all these sort of energetically demanding tasks, that the orange individuals kind of wear themselves out and therefore aren't in a good position to survive the winter and make it to the next year. The other argument, and it's kind of implicit in here, I don't think I wrote it up here, but again, testosterone oftentimes can inhibit immune function. So if you look at inflammatory responses, for example, how much you swell after you get an immune challenge, individuals with high testosterone tend to not have as strong an immune system. They don't have as strong an inflammatory response. And that also can result in lower survivorship because you just don't have the ability to combat diseases or challenges with your immune system. The last axis is this one that I want to talk about. I mean, there are others as well. There's one involving the thyroid gland. There's some that are related to growth hormone. This one is called the hypothalamic pituitary adrenal axis, or the HPA axis. And if you've taken much psychology, there's a chance you've talked about this one because this one seems to be really important in humans, especially when you start talking about anxiety disorders and other kinds of mental disorders, that this axis can be involved. And the reason, in part at least, is that this is basically our stress response axis. So the basic story, I'll do this pretty quick and then we'll break, is that again, the hypothalamus releases a releasing hormone. This one's called corticotropin releasing hormone. It travels to the pituitary gland, which then releases its hormone. This one's called adrenocorticotropic hormone, or ACTH. And then ACTH travels to the adrenal glands, which in humans are perched right on top of our kidneys, and causes them to release glucocorticoids. In the case of humans, it's cortisol for the most part. Other animals, it'll be other variants of these kinds of things. But they're all these stress hormones. And the job of these stress hormones are to upregulate processes that allow us to handle stress, be it like emotional stress, social stress, physical stress, trauma, there's not enough food around, these kinds of things. It triggers all sorts of responses that are related to those kinds of stressful events. So they can be acute stressors or they could be potentially chronic stressors. That will then alter the behaviors of the animals. So that's where we need to pick up on Tuesday is how these things will alter the behaviors of animals. I'm sorry we didn't get to the homework again. I think I'm probably going to take a break from homework on the next chapter to let us kind of get caught up, but also, you guys are going to be working plenty on the homework, on the exam rather. Okay, so have a good weekend.